part of the phylum. So you guys, we're going to talk about what's in the Cnidaria club. And anytime you see a P up above a word, that means the phylum. Okay? C would be class, and Bill would tell you that there's supers and subs, and there's like 20 different levels of organization. But in the Cnidaria club, we have the sea anemones, the corals, the sea pens, the jellies, jellyfish, and the hydrates. Now that's the big group that's in that phylum. Those are the three. The C's they're all classes. Okay, and that's on the as you can see on this chart that we made a long time ago. We're way down at the bottom of the tree, or the tree of life. We're just going to talk about the sea anemones, because that's what you're going to get asked the most questions about. That's the elephant in the center of the room and some of the touch tanks, the big green anemones. And what I'm going to say about the anemones will apply to the jellyfish and the hydroids, things that you're not going to get asked so much about. But Bill had a great lead-in. It's where did the Cnidaria fit in that tree of life that we've got here? If you look, they're over on the down in the bottom left corner, and we use the same kind of a stick drawing, and this is pretty much a traditional tree. And we thought that the sponges come off first, and then the Cnidarians, and these are the Ctenophora, the comb jelly, and then you go up here to the bilateral, all the other animals. Well, everything changes. And from when Bill and I did the last invertebrate taxonomy talk, things have changed, and it's based on DNA. Okay? What they've done is it looks like the Ctenophora, the comb jellies, may have branched off. Now we're talking like 600 million years ago. Branched off very, very early, and then as you go up, the sponges may have actually retrogressed and lost the muscle and the nervous tissue, and so they've been moved a little bit up. And here's another, and this is all fairly new data. Here's another thing I took out of science just in the last fall. If you start out down here, they gain a nervous system, they gain a mesoderm, and you've got Ctenophora over here. The peripheral, again, looks like they've de-evolved. They've lost the nerves and the muscles. Placozoa is an interesting little phylum. Probably more people are making a living off of it than their species. There's one species of animal, and it's so different, it's its whole phylum. Okay, so we never really talked about it, but that's what that is. And then you've got the Cnidarians and go up here to the bilateral. So we have basically changed the way the tree has been made. This is the new tree again. But the big thing I'm going to say is that the Cnidarians were down at the bottom of the tree, way down low below everything else. And so there's the comb jellies, the Ctenophora, which I love, sponges, the Cnidarians, and you go on from there. So the key points about the anemones, or the Anthrozoa, there's three key points. They all live in the marine environment, and they're all carnivorous, sort of like Bill. They are they have nitisites, stingers, and these guys have tentacles. Okay? Does anybody remember the term solentorata from when you were in school?
That was going to be my first question.
Solentorata was what we used to call Cnidaria. Yeah. So that term has changed. So things changed. When I first got here, Bill was talking. I said, "Well, where's the solentorata?" You know, because I'd been in medicine for all these years. Didn't really. I was behind to catch up. I didn't know about the change. But anyway. The nitisites are such a big deal that we basically named the phylum after it. And so we're going to talk about these guys. Okay, the anemones don't have a polyp form. The jellies have a polyp form, but the anemones, I'm sorry, they don't have a medusa form. The anemones do not have a medusa form. They are always on the bottom. That's where they are. So they don't go floating around. Okay, so that's a fairly significant point. Okay, Bill let me talk about attachment and movement. Bill and I talk a lot about these kind of things. And the question is, how do they stick on their rock? That's basically they do it. And so I spent a lot of time getting ready for this talk about 3 or 4 months ago. We did this attachment talk about 5 years ago that we'll talk about that again this afternoon. But basically, I couldn't find any evidence of a biological adhesive in the literature where the Cnidaria used it. So there's this lady named Daphne Falk, and she's one of the world's experts in sea anemones. And what university do you think she's at? She's in Kansas. Okay? So this shows you don't have to be right by the ocean to study something. A lot of people study things on the other coast of another continent. But basically, I emailed her and said, she'd written a lot of stuff, and I'd read a lot of it. I said, I can't find out anything about how the anemones attach. And she said, good. So she wrote me back a really nice letter and said, it looks like, even though they're a snotty little animal, they've got mucus all over the place, but they don't use a biological adhesive. It looks like it's suction. And there are certain anemones that attach to hermit crab shells. And so when the hermit crab moves to a different shell, the anemone detaches and goes to that shell, or sometimes the crab will help the anemone move. So there's some sort of, and I haven't gone into this more, it's just a matter of time, I guess. But anyway, so they attach by suction, and they don't have an adhesive. Yeah.
The gecko can walk on glass because of the molecular attraction between their pads and their feet and their substrate they're walking on. Is it possible these guys do the same thing?
No, we're talking about that this afternoon. Yeah, exactly. That well, I've got pictures of all that. So when they move, they do two things. They can either do inching along, like an inchworm, or they can somersault. Or what they do, and I'll show you this a little bit later, what they can do, they can detach and they can roll down a slope underwater if there's enough slope. So they've got several ways, but that's, so how do they reproduce? Well, you guys know this. They can do it several ways. They can bud, and the brooding anemones do this, and then they can pull apart, and we've seen this happen in the different tanks, especially the wall tanks. Or they can use sexual reproduction. And here, this is again broadcast spawning. Basically, they throw the eggs and the sperm off. It forms a planular larva, which floats around and settles and becomes a new organism. And that's the way, if you're going to move your range a little bit, that's the way, if you're just separating by fission, you're not going to go very far. So the general layout of the anemone, this is really this is up by our touch tanks, and this is pretty good. This is, for our purposes, this is great. This is all you need, and more than most visitors will want to know. So I don't have much to add about that. That's really good, but I think something that's really interesting is how the anemone keeps its shape. And that's what we call a hydrostatic skeleton. Okay? And this is what we mean by the hydrostatic skeleton. It uses water pressure to keep its shape. The symphonoglyphs they come down the pharynx. The symphonoglyphs are two lines of cilia that wave and push water into the center of the anemone. Okay, so the water comes down that way. Okay, so that's what inflates the anemone. It deflates by the use of muscles. There's two sets of muscles. There's actually three, but there's a sphincter muscle around here that's like a purse string, and it can contract that. And there are retractor muscles along the septum. So if you go out at Seal Rock or someplace when the tide is out, or they want to, they're really little and they're all shrunken down. Some of ours will do that too, but not to the degree they do it out in nature. They really can hunker down. And then there's another muscle around the base of the foot that's probably involved with motion. And this is from Daphne. I haven't really looked at that too much. But anyway, that's called the hydrostatic skeleton. That's how they keep their shape. Okay.
The water that brings in this is a constant.
Yeah. And there's some, I'll show you, there's some little pores. We'll go back.
Yeah, I saw that.
Yeah, there's little pores that that the water gets out through it. I've got some more pictures of that in a little bit. Yeah.
Just real quick, many of you have probably seen that really, really large white anemone that's in the eye level tank. Doug, how often have you seen that collapse all the way down?
I have only seen it once, and somebody said, gee, it looks terrible, and I looked at it and went, wow, I've never seen it like that. It looks terrible, and talked to Colleen about it. She said it does it at least once a week.
Yes.
It was that way Saturday.
Yeah, and I don't know how often it happens, but it's kind of interesting. I wanted to feed off what you were saying, because the green anemones don't seem to go down like that, and the pink tip ones do, but that white one really seems to do it, and it's not because of tides, because it's still in the same spot.
And I have never seen either of the big white ones in Bird's Eye or the ones that used to be in NOAA do that either. And for all I know, they do it just as frequently as the one in eye level. That's the only one I would ask Colleen about.
So one of the things you can do at the touch tank is to point out, everybody can find that white anemone. And once they realize it's an anemone, an animal, it pretty much sticks about the same spot, so you can direct them back to the large tank behind that has the real big white anemone, which is the island.
Good. The Nitisite is the main cell, basically, that the phylum is named for. This is the workhorse of the anemone. And the Nitisite consists of receptors here, and then the little harpoon trigger that it uses for defense. So these are mechanoreceptors, and I was going to put electron microscope pictures in there, but we don't need to. But basically what happens is that these get stimulated by vibration, and they cause the harpoon, the stinger, to shoot out.
The stickiness then is you're being harpooned.
Right. The sticky, there's little barbs on it, and all of the nidicites in different organisms and the different classes in Cnidaria are a little bit different, but they're basically all the same way.
I better not tell them that.
Right.
There's some little kid, oh yeah, that thing's harpooning you.
Well, they're not going to get through our skin, but a little tiny baby, you tell, it would get through their lip or something, so, we don't want to kiss them.
Yeah.
The thing is, yeah,
How much variation is there in them? I mean, there are jellies that you can't even feel, and jellies that will put you in the hospital for a week. Are there anemones other places in the world that pack a punch? None of ours seem to.
that have the,
That have a real kit when they sting you.
yeah, we're going to talk about that a little bit later, but yeah, there's a lot of variation, but the toxin I'll tell you about in just a bit here. The thing about the anemones, when they shoot off their nematocysts, it takes a lot of energy, and it's been estimated it takes 25% of the, in order to catch a brine shrimp, this anemone the hydrate has to shoot like a quarter of its bullets. It takes 48 hours to regenerate, and so they only want to use them when they have to.
What's the difference between the nematocyst and
What?
what's the difference between the nematocyst and the Are they the same?
The nematocyst is the harpoon in the nitisite, which is the whole cell. And the nitisite has in it, remember what I said, I showed you that little picture of everything sticking up. This is a blow-up of that. And there are, because it's so expensive energetically for the anemone to capture something, it doesn't want to waste its bullets, I guess, or the stingers on things that don't count. So it turns out that there are N-acetylated sugars on the surface of most like larvae and prey that the anemone would eat. And as they swim along, these sugars wash off, and the Nitisites recognize this as a chemical signal. Hey, there's a prey here in the area, I'm going to get ready to shoot. So that sensitizes the Nitisites to shoot, and the more sugar that the anemone sees in the area, the more they discharge up to a point. And if there's just a huge amount of sugar, I'm making this up sort of, but I think I'm right, is that probably that whatever it is too big for them to catch anyway, so I'm not going to waste my time on it. So there's a curve, and I basically made this up out of some research papers, so because the real curves are like hard to read. So swimming prey goes by. Here's a little larva, okay? And there's a lot of things under the microscope that look about like that. That's a brine shrimp larva, but copepod larva, and all these other things look about the same. And these guys, when they're swimming, they set off a radiation, and that vibratory sense is really, really, really low. That's 2 to 30 hertz. We couldn't hear that low, okay? But the anemones, the Nitisites respond best in that low range. What a coincidence. Okay, these guys have had years to evolve, and so they have learned that, yeah, when this vibratory sense is in that low range, there's probably something we want. There's not too much sugar coming off. So they've got this thing worked out. But they've only had like 500 million years, right, to do it. Okay, the other part of the Nitisite is that the wall is, I want to talk about the wall because that's pretty important as to how this all happens. The wall is reinforced with collagen. And the wall is really, really, it's a tough wall, a tough surface. And there's a Nitisite coming out. I'm going to go on about this. So the question is, what makes the Nitisite, how does it shoot out? How does a stinger actually shoot out? What is the physical chemical nature of that? And it's been proposed that maybe it's like tension, like a spring-loaded thing. Maybe it's contraction, that the wall contracts and pushes it out like squeezing a pimple. Okay? I guess that was not a good choice. Or osmosis. Is there anybody that doesn't know what osmosis is? Basically, it's the passage of a liquid or a gas through a semi-permeable membrane for an area of high concentration, low concentration. Well, what happens is that this works. Okay, this is the mechanism, and you've got all these great big molecules in the center of, in and around the Nitisite, and when the sensation, when the stimulus comes to shoot, these big molecules dissociate. They break up into small molecules, and that increases the osmotic pressure, and water rushes into the cell and pushes out the stinger. And this happens incredibly quickly. The pressure goes to 2,000 psi in 2,000th of a second. Now, 2,000 psi is a lot. We run hydraulics on John Deere's and Caterpillars on 2,900 psi. That's how we raise the lower blades. 2 milliseconds is really, really quick. So this all happens just in the blink of an eye. Now you think, well, 2,000 psi, that's a lot of pressure. That's pounds per square inch. We're not talking square inch here. We're talking tiny, tiny little amounts of surface. So that 2,000 psi does actually, it's achievable and it works. Okay, now the toxins are called anthroplurines, anthrozoa, anthroplurines, and these are toxins that actually paralyze things. And in 1902, they did some studies where they put they ground up anemones and they gave dogs the anemones, and all the dogs died. They had heart attacks and they just stopped and they died, and so that experiment probably was hopefully reproduced. But basically, what the anemone toxin does is it blocks the sodium-potassium pump. and we have a sodium-potassium pump that keeps our sodium at a high level in the cells and the potassium at a low level. If that pump doesn't work, we're done. We have a heart attack, we're dead. This anemone toxin blocks that. It's very similar to scorpion toxin, but probably 110 times stronger. They studied this quite a few years ago in the 60s and 70s to see if it would have any medicinal effects. It does not. It's just the toxin. So there's no medical use for it. Talking about the defenses and the predators, and I'm trying to give you guys stuff that you can give to the public. This is really interesting. This is the Anthropologism elegantissima, or the aggregating anemones, and we have these anemones here. And basically, these anemones, most of the time, divide by fission. So they're all genetically the same. Okay, so you've got one whole group of anemones that's exactly the same, and a little different group over here that maybe goes back, hundreds of years or a few years, but they're a little different. And these guys don't like each other. Okay, they're very agonistic if if their genes aren't exactly the same, and they wage battles. And some of their tentacles have remember back in the Middle Ages they had these balls with spikes on them that they swing on the chains and battle. Yeah, that's what they've got these tentacles that are modified that way. And not only is that, but think about this. These are very, very simple animals. And this clone is broken up into scout. There's scouts that are out here in no man's land, which I'll show you. There are scouts. There are warriors that have the battle tentacles. There are free edge polyps. There are the reproductive ones that are in the center, away from all the action. And they have reserves. So they're basically like an army. I think that's just amazing. The other thing they do is that they fight at high tide. So low tide comes, there's no water. It's like maybe in George Washington's time, the armies all took the winter off, and then they go back to having war in the summer. And so the anemones fight at high tide, holding their area. And these guys, with these attacks, they can peel off. And the ones that get it the worst are the scouts. Okay, they just wail on the scouts. And if the scout is beaten up so badly by the other side, they may not recognize it as one of their guys, so they start wailing on it too, and the scout doesn't. So you don't want to be a scout if you're an aggregating anemone in a colony. You want to be big. I pick the reproductive one in the center.
I have a question.
What?
I have a question.
Yeah.
Do we have warring groups in our touch pools?
We don't have any in our touch egg, but they're certainly up and down the coast here.
Okay.
If you just go back, did you tell about the line of the DMZ?
What?
The DMZ.
Oh, I've got another picture of a better one coming. So these are the clubs, and here's another set. Here's the DMZ right here. That's really, really apparent. Okay, that's almost like North and South Korea. Okay, and here's a little scout. He's out there, and here's closer to and here's some of the anemones. They're leaning forward, wailing away with their tentacles, their agrophoria. So this is really, I think that's an amazing story to be able to tell, because these guys are the lowest, they're pretty well down there on the evolutionary tree, but they're still organized, and they have separation of activities.
If they wage war, is there any hope for us?
Yeah, probably not. All right, so what else? So what eats anemones? Anemones are pretty tough. They've got this toxin. Well, there's a nudibranch that's really good at eating. We have these nudibranchs, and I've actually got photographs. These are off the internet. These are better than mine. These are all the same one. Okay, so what happens when the nudibranch comes up to, and a bunch of anemones. And this was done with the aggregating anemone, and this was from about the 1970s. So here's the nudibranch coming up, and these are the seconds into the study. The nudibranch comes up and touches the outside anemone, and the anemone, I mean, will probably take a bite. The nudibranch would love to eat the tentacles. That's the thing it really wants to eat, okay? So, but the anemone, the aggregating anemone, said, I'm not going to let this happen. So he puffs up and pulls back like this, gets up and away, and by doing that, he gets just tall enough that that nudibranch can't hardly get to the tentacles. Well, the nudibranch then takes a bite here, and he goes on, he says, can't get to the tentacle. Maybe there's somebody that hasn't puffed up yet. So he goes to a different one. Well, that's good, because the anemones can take 1 or 2 bites on their stalk, and it's not going to hurt it. But if the nudibranch just keeps working on one anemone, he's probably a goner. So they can fight back sometimes, but they usually deflate or withdraw, and they can puff up. Remember I said that if they're on a slope, they can puff up and roll downhill. Well, all this stuff happens in slow motion, basically. The nudibranch doesn't go very fast. The anemones don't go very fast. So if you roll down the hill even a few inches, you're probably safe. So anyway, the other thing that happens is they've got this toxin. Well, the nudibranch has evolved so the toxin doesn't hurt it. In fact, the toxin helps the nudibranch because it gets under the skin and nothing else wants to eat the nudibranch because it's got this toxin. The problem is, the story gets better, the problem is the toxin is leaking into the water. And the anemone, and so it's like the nudibranch's broadcasting that, hey, I'm coming for you, because here's the Anthropologism toxin. The anemones all smell it. How are they, their olfactory receptors get. They see that they're toxic. Here comes the nudibranch, and they all go into defensive positions. So it's a whole very complex situation, because the Anthropologism helps the nudibranch, but it also warns the anemones. This is all at the bottom of the food chain. This is just, to me, this is just amazing. All right, the anemone's got one more trick up its sleeve. Okay, here comes the nudibranch, and or something else, and that's these little synclides, the little perforations here, and it's got some little hairs called acanthium that it can stick out through these little pores. Now, if you happen to step on an anemone in the ocean, and you step on it, seawater goes every which way, doesn't it? Think of those little holes as like a pop-off valve. Okay, it prevents it from getting injured. But if the anemone is defensive, here's a picture of one with its acanthium stuck out, and all these guys have Nitisites on them too. So it's like in a medieval castle. If you're trying to storm it, they've got little windows they can shoot, the guys coming up the ladder. So these guys, even though they're low down on the scale, they've got some tricks. Mosshead sculpins are, other things that eat anemones are the sculpins. They don't seem to, the Anthropologism don't seem to bother them. They eat all of ours. These are the different ones here that we've got. They'll eat any of these. The Leatherstar, there's another picture of that. The Leatherstar eats anemones. And here's one that's about 400 feet down in the ocean. This is an ocean cable. And on the cable are anemones, and the Leatherstar is wrapped around the cable eating the anemones. So that's something that I won't see because I don't go that low. Okay, then we've got, the other thing is the Endosymbians. And there's two sorts, and Bill loves this, the zooxanthellae and chloraxanthellae. Zooxanthellae are dinoflagellates, which are their own phylum. This is what's responsible for red tide. And they live in, mostly in the endothelial cells of the anemone, so on the inner surface. They generally are brown in color. There's a lot of species, and more than one species can be in the same animal. They tend to occur where there's a little bit more light interestingly, anemones that have the zooxanthellae move toward light, and ones without it don't. So there's an interaction here between the endosymbiont, dinoflagellate, and the anemone. The anemone moves toward light to try to help out its endosymbiont, which is fine because I'll show you a picture of this a little bit too. So here, dinoflagellates, as you know, that they're in a different kingdom actually, even red tide, and they've got the little tiny flagellum, and that's how they get around. There's a variety of species. I've never really looked too carefully at dinoflagellates. I don't know if you've done any work with them.
The sea foam that you see right now
Right.
is basically dinoflagellates. It's their skeletons sticking in the water together, kind of like soap.
Yeah. The other thing that's in the anemones are these Chlorella. And this is a marine algae consisting of Z. parasitica. Here, there's only one species. Okay? And this is basically more green, and this can live in the same anemone with zooxanthellae. They can be in there together. I said there's a lot of species of zooxanthellae, the dinoflagellates. There's only one species of the Chlorella. And this will do better in even lower light than the zooxanthellae. And they both help their host by fixing carbon and transferring it. They take up the host waste products, the carbon dioxide. The zooxanthellae is better at that than the Chlorella is, but it's, they both will do the same thing. Blanching, and you guys have heard about this. This is the loss of the zooxanthellae and the Chlorella. It can be due to acidification, pollutants, temperature change, or whatever. And this, you can reverse it if it's not complete.
Yeah, this is mostly with coral. You've heard this coral bleaching. The same idea with its
Right.
temperature gets up.
Same thing. Okay, the last thing I want to talk about is the anemone fish. And this little clownfish lives in the anemone, and this is he gets by because he's covered with a mucus that protects it from the anemone stings. The problem is that the mucus is species specific, so if he goes to a different species, he's going to get stung. Okay. The other thing, and John Chapman I was going over this with John, and he said he put some clownfish in with an anemone, and the clownfish was going to go and say, "Hi, anemone, here I am." Well, the anemone stung and ate it. And so John had to go buy some new clownfish and introduce them a little bit more carefully. If the clownfish gets by the initial contact, it starts the mucus. But if it just runs up to the anemone, all happy and everything, it's going to get stung.
So a mucus-covered fish is a happy fish, and so when you move fish around, like you buy them from the aquarium,
Yeah.
they stop the mucus production. So it's also the case where they don't realize that they don't have the mucus to protect themselves. So what we do is we isolate them, we keep them in bags, and let them basically quarantine themselves, and then give them a chance to grow their mucus back.
So the clownfish are basically there. The anemone gives it a place to live, but the clownfish protects the enemy. They've done studies, and we're talking a little bit more tropical now, where they take the clownfish out of the anemones. Within half an hour, the butterfly fish are nibbling on the anemone. So there's this fairly significant symbiotic circle where the anemone fish produce ammonia that's taken up by the anemone. The anemone has zooxanthella, which is useful chemically. The anemone protects the anemone fish. The anemone fish protects the anemone. And it's just this multi-layered symbiosis. All right, with that, remember, the enemy of my anemone is my enemy. So that, and then, so I think we're pretty much still on time. Yeah, let me turn lights on.
Just a quick question on the interaction of a person and two anemones in our touch tanks, given all of this. So do we have too much sugar on us and they back
Do
away?
we have what?
More sugar and we're large, right?
Our skin is just too thick to get through.
But they touch us. I mean, we can feel,
Yeah.
people under understand what's
The
happening.
other thing is our poor anemones here get touched so much,
Right.
they pretty much are out of bullets. They're out of stingers. If you go down to Seal Rock and touch an anemone that's in the tide pool there, it's way stickier. So I think it's just a matter of captivity. Bill?
Well, I think this is another good point of interpretation. And you know how we always tell them not to touch the oral, the mouth, which is also the anus, which always chews them off anyways, but touching the sides rather than the tentacles themselves. I'm always a little nervous when I see a mom or a dad with a baby touching the tentacles. And the reason I am is because this is a true story. There were some 6th graders that were there, and I was telling the story behind the touch tank, and I said something to the effect that it's only because our skin is thick, and that if you kiss the anemone, that you feel it like a jellyfish sting. And the one boy bet the other boy wouldn't do it, and he really did it. He kissed it, and his face got red and swollen. So it definitely had a reaction. And it was just because, I think because the skin on the lips was very thin. I don't point that out anymore, you know, this is, but I do know, I mean, that did happen.
So essentially, they're seeing us the
Well, and it's kind of individual too, because I've seen any number of, several month-old babies have their hands
me
put
too.
under the anemone tentacles, and it faces them not at all.
Yeah.
I met a strapping young man in his early 30s, 6'2", as fit as he could be, at Yuquina Head, and he said he touched the anemones and he was numb for 3 hours after.
So he was probably very successful.
Individual variations.
Yeah. No kissing anemones.
No.
No.
Yeah.
I have people ask me, when you feed them, how long it takes for them to digest a piece of food. They happen to know.
It goes, it's a matter of minutes to hours. We did a study here in this lab about 5 or 6 years ago where we got guppies from Walmart, and we had a big green anemone on a rock, and we fed that anemone a guppy, and it basically was hungry. It stung the guppy and stuffed it down. We waited 5 minutes, gave it another guppy, stuffed it down. It went through 14 guppies before it got so tired and so full. Then the first string all closed up, and it didn't do anything the rest of the day that we could see. So these enzymes down there are fairly powerful, and I've read at one point that if you stick your finger down there and hold it for 20 minutes in the anemone, and it's that when you bring it out, you'll be partially digested. I haven't been able to find that recently, so I sort of quit saying that.
Another really good explanation, because they're right there side by side, is I've seen green anemones that ate a purple sea urchin and then spit the test back out again. So they dissolved everything. I mean, I didn't see anything other than the test coming back out.
What's the text?
That's like the skeleton, the round thing underneath. It's really pretty.
Here's one.
Yeah. Underneath all the spines.
There's a video online of like a lion's mane jellyfish, one of those giant ones bumping into a wall, and the anemones could just suck that thing in and ate it.
The anemone did.
Yeah, it was huge.
They're tougher than you think.
I mean, the whole wall, they just dissolved it in parts.
Here comes dinner.
So I had a question about those leatherstars eating the anemones, because in our middle high school last week, we had 3 leatherstars eat some of the aggregating anemones.
They did.
They did. We saw them do it over a couple days. Do they just eat the aggregating anemones? Will they eat any of those anemones, or?
I don't know for sure. My feeling is they'll eat anything they can catch. That's usually true for most things in the ocean. When somebody eats this or that, most things are pretty generalist in their diet.
When you feed the leather stars,
We give them, lots of people give them mussels. I try to give them something else after I learned that they actually ingest stuff, so I try to give them things that are easier to swallow. But that's the other thing is, in our environment, everybody gets fed all the time, and so they're not so likely to be eating their tank mates.
Or they eat that aggregating anemone?
Well, he was looking really sad already, like
Yeah, but
he
you
was
think it's
not,
impossible because
right.
he was splitting, but.
So he wasn't very, he was obviously easy hanging fruit.